like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loonin School of Medicine Healthy Longevity webinar. As always, thank you for joining us. Today we have a very important topic. We are talking about ovarian aging. Professor Eva Hoffmann is with us tonight, and I will introduce her in a few minutes. If you would like to learn more, even more than today, about ovarian aging, you might want to consider to attend the Asian Centre for Reproductive Longevity and Equality Conference, and that's going to happen at the 14th and 15th of September 2022. There will be a link in that chat box to find more information. Another very important, dear to my heart event is that you should not miss the opening of our Centre for Healthy Longevity. And that's going to happen at the 7th and the 8th of September. And there are two different programs. On the 7th, we have a hybrid program, so you can join virtually, but also in person, with, I would say, top international researchers, including, to name just a few, Matt Kabeline, Professor James Kirkland, Professor Nir Basilai, and Dr. Felipe Serra, and many, many more. On the 8th, there will be a face-to-face community outreach event at the Alexandra Hospital, where our Centre for Health Longevity, the clinical site, is actually placed. So also for both events, please join and share the invite with your network. And we are really looking forward to welcome you. And as always, the registration is for free. You might realize that this is a very, very busy week for us. So due to all the events that week, we will not have an additional webinar on the 8th of September. But therefore, we really invite you to join the 7th and in a hybrid way, or the 8th. For today's meeting, as always, please leave your questions and your comments by using the Q&A function. We have a short presentation today by Elisa Marie Crombie. She is a PhD student from the Dr. Zeiss Aging and Metabolism Research Lab. And she will talk about how to rescue mTOR-C1-induced sarcopenia. And sarcopenia is a low muscle mass occurring during the aging process. Elisa, the floor is yours.
Thank you for the introduction. As mentioned, my name is Elisa, and I'll be presenting my paper entitled Activation of ERF4E Binding Protein 1 Rescues mTOR Complex 1 Induced Sarcopenia by Expanding Lysosomal Degradation Capacity. Sarcopenia is the age-related loss of muscle mass and performance, where aging is the primary associated factor. Multiple secondary factors also contribute, including inactivity, disease, and malnutrition. Exercise training is currently the only known intervention mitigating sarcopenia progression. Therefore, it's important for us to identify molecular mechanisms involved in sarcopenia to identify key therapeutic targets. mTOR complex 1 is central to skeletal muscle aging, and as a protein complex is regulated by nutrients, phosphorylates SHK1 and 4BP1 in the regulation of protein synthesis. In aging, mTOR complex 1 activity has been shown to be hyperactivated, associated with reduced lifespan as well as reduced muscle strength and mass. Rapamycin, as an inhibitor of mTOR complex 1, has been shown to extend lifespan and improve sarcopenic features. Therefore, we wanted to identify how could reducing mTOR complex 1 activity through SSK1 or 4BP1 improve sarcopenia. To do this, we utilized the TSC1 muscle knockout mice. In this background, we additionally knocked out SSK1 or activated 4BP1 by mutation to identify specific roles of these mTOR complex 1 targets. We found that only 4BP1 activation was able to rescue deficits in muscle size and strength seen in TSC1 muscle knockout mice. Similarly, myopathy was present in TSC1 muscle knockout mice, shown by pathological features of the muscle tissue here. This was only rescued by 4BP1 activation. Another pathway involved in aging is autophagy, which has been shown to be defective in aging muscle. Autophagy is a cellular degradation pathway where misfolded proteins and damaged organelles are degraded by the lysosome. mTOR complex 1 is known to inhibit this pathway. We found in our study presence of P62 and ubiquitinated protein aggregates, indicating defective autophagy function. This was rescued by 4BP1 activation, but not SSK1 knockout. We found that these P62 protein aggregates were accumulating at the lysosome, indicated by colocalization of the P62 signal with the LAMP1 protein. By ultrastructural analysis of the lysosome, we found that lipofuscin aging pigment was accumulating within the lysosome, which may inhibit the degradative lysosome function. An enzyme present within the lysosome is a cathepsin L, where the mature form of the cathepsin L protein indicates the active form. The active form was shown to be overexpressed in the 4BP1 mutant TSC1 muscle knockout mice, indicating that these mice may have enhanced lysosomal degradation capacity to clear the protein aggregates. By magic red staining, we confirmed cathepsin L activity. By lysotracker red staining, which is an indicator of the acidic lysosomes, we found reduced lysosome activity, which was rescued by 4BP1 activation. Therefore, we conclude that 4BP1 activation enhances the lysosomal degradation capacity, clearing protein aggregates and delaying the onset of sarcopenia. Thank you for your time.
Thank you so much, Elisa, for this great work to really finding a cure for sarcopenia. I would say in the meanwhile, let's all go to the gym and restore our muscle health and maintain our muscle mass. Muscles are not the topic of today's main presentation. The main presentation is by Professor Eva Hoffmann. And she is a professor of molecular genetics and the head of the Department of Cellular and Molecular Medicine at the beautiful University of Copenhagen in Denmark. And her research really focuses on reproductive genetics and how that is regulating longevity and especially ovarian aging. I would say, Eva, it's a great honor to have you here. We would like to learn everything about reproductive longevity and ovarian aging in women. The floor is yours.
Thank you very much for the warm welcome. And it's really wonderful also to be interacting with Southeast Asia, and specifically a topic that is so important worldwide. I'm going to be talking a bit about the work that we're doing on reproductive longevity, and specifically also on trying to understand the regulation of why we differ, why we vary with regards to ovarian aging. So the way that we kind of looked, and I think at the picture at the moment with regards to female reproduction and particularly also ovarian aging, this really falls under the larger umbrella of women's health. We know that there's very little funding historically that's gone into this, and we're hoping that obviously this will increase because we believe that this is an area of critical importance for several reasons. But we also know that about 1 in 4 women's visits to general practitioners are for female-specific problems. And nearly 1 million women in the UK have left the workforce because of problems or symptoms associated with menopause. 7 out of 10 women that our collaborator Anna Murray surveyed in the PPI at Exeter University, they were actually unaware that infertility is likely to decline a whole decade before menopause onset. We're also seeing an increase in IVF, about 30% increase so far in the past decade. And here in Denmark, where we have IVF free at the point of care, 10% of our babies are born after medically assisted reproduction, 6% from IVF and ICSI, and the other remaining from intrauterine sperm injection. So trying to understand reproduction and ovarian aging and how it influences not just fertility, but also other phenotypes, clinical phenotypes, is one of the, I think, main topics and challenges that we are faced with now. And the reason, of course, is that we've seen a great increase in our lifespan, also health span, the past century. But our fertility curves and also our natural menopause curve really have remained similar and static around 50 to 52 years for menopause. And menopause is the cessation of ovarian function. It's when our ovaries have been depleted of oocytes, and it's an endocrinological phenotype really, where estrogen levels start falling afterwards. We know, though, that these two curves that we're interested in are seen across the world. It's not just a Western phenomenon, it's not just an OECD phenomenon. This is a phenomenon that we've seen over centuries. And in black here, I'm showing you the fertility curve according to female age on the x-axis. On the y-axis, it's just the proportion of women that have been surveyed. And you can see here that what we see is that around 30 or late 20s, we really have a nice high peak of fertility that then falls as we age. We also then start seeing a different peak, and that is an onset of natural menopause. We know that for both of these, that they are associated with aging phenotypes or clinical comorbidities, and I will touch a little bit upon that in my talk as well. I'm not going to focus so much on the fertility curve. I'm happy to take questions on it. We know that the fertility curve seems to be really modulated by chromosome errors in human eggs, the eggs that we ovulate as we get older, but also in the younger women. We also know that clinically, if you have a diagnosis and you look at those women who have recurrent pregnancy loss, that means 3 or more consecutive losses, either primary or secondary, that 10 years later, we start seeing other comorbidities appearing. And it's a whole plethora of range of different clinical phenotypes. They include type 2 diabetes, but also psychiatric disorders. With regards to natural menopause, I think we all understand that hormone replacement therapy is one of the ones that are being used. For example, if there's a history, family history of osteoporosis. But there are also a number of other comorbidities that are appearing, suggesting that this distribution of when we go into menopause might be very important. The way that we kind of look at this, of course, is that these are distribution, and this is something that all females go through. So this means that we're looking at a multifactorial phenotype that we have to try and understand. And that's what we try and do in our lab, is to try and understand the biological mechanisms that shape these curves. And we are hoping, obviously, that this will lead us to refinement in terms of the molecular mechanisms and pathways, and eventually also a personalized approach to diagnostics, prognostics, and potentially also interventions. So I'm going to talk about work today that we do as part of a much larger consortium. This is from a recent paper that we published. I'm going to basically merge all of our our data here, and it was published last year in Nature. And we also have a second follow-up study that you can see down at the bottom here that's now deposited on medRxiv. And basically, we have 5 leading centers here, and then we have hundreds of different other co-authors and colleagues from individual studies who contribute data and analyses, because we really want to understand these phenotypes, not just in the UK Biobank, but also globally. So first of all, how do we actually look at ovarian aging in terms of menopause and in terms of its function? Well, we know that during development, fetal development, total oocytes, they about 6 to 8 million. This is during the fetal development of the female fetus. And at birth, there's a great depletion. Upon puberty or menarche, there's another great depletion, and that continues until the onset of menopause at around 50 to 52 years. At menopause, we define this by having about 1,000 oocytes left or follicles left, but they don't seem to be functional. They might be senescent. What we see as well is, if you look at the actual life history of these follicles, these specific cells, is that we see germ cells that go into meiosis, which is when we start seeing the reduction in chromosome contents, and they then form follicles with somatic cells and then start resting. So at this stage, at birth, we're basically looking at the establishment of the ovarian reserve that has to last females for the rest of their reproductive lifespan. There's attrition, as I said, at all of these stages, but at the onset of puberty and menarche, once there's continuous recruitment, and once the onset of menarche with the HPO axis and the hormonal recruitment and selection of follicles, they can grow, and one follicle per month basically ovulates a mature egg that can be fertilized, as shown here to the right. So what actually determines how many eggs we have, how many eggs we actually ovulate, and the depletion rate over time? This is obviously just an average curve here, estimates from cross-sectional studies. So what we're interested in is trying to refine what determines and regulates menopause as an endpoint. And I've just put it in here to just show the follicle numbers as we see them. It's the follicles that are growing that are the endocrine tissue type, basically, that produce estrogen. And you can see here that we start off high and then we go down again. And at the most fertile, we have quite a few oocytes still left, although, of course, we are on a log scale. The fertility decline really starts happening in the mid-30s, both with regards to the loss of follicles, but also with regards to the quality of eggs that are being ovulated. So the mission and objectives that we have as a consortium is really to understand the biological mechanisms and the genetic architecture that govern ovarian establishment and its depletion or aging over time. We're interested in the impact on health on menopause timing. Are there basically certain phenotypes that we can identify that are associated with when, if early or later time? We're also interested in using genetics and other factors to predict reproductive lifespan. And that's obviously very important with respect to fertility planning. And then finally, we believe that if we can understand the biological mechanisms and also the genetics, it may help inform treatments, diagnostics, therapeutic approaches that may allow reproductive longevity and improve fertility treatment. So focusing on menopause now and the endpoint as ovarian aging, we know that this is a distribution and it has a tail very low that clinically below the age of 40 we define as premature ovarian insufficiency or primary ovarian insufficiency. But you can also see here that this is self-reported from the UK Biobank. This is good enough, we know, because we have large numbers, that very clearly that it varies. And some women, they experience menopause at 40 and others at 60. And that's what we're interested in, understanding the population variation and how that's regulated. We also appreciate, and we've appreciated for a long time, that both genetics and environment contribute to differences in age at menopause. I believe that's very similar for any other aging phenotype. we've recognized for quite a while that the mom's menopause age is very important for her daughters. We think that at the moment that about up to 50% is genetically determined, but there might also be interactions with this with environmental factors, for example, smoking and other things. When we try to then tease apart the genetics, which I will now focus on, this is things that obviously that we can now start addressing because of the revolution that we've seen in genomics. Traditionally, what you see here on the right is that we look at allele frequency in the population. And that means basically what kind of genetic variants do we see. So if they're very, very rare, they're usually something that we see confined to families, and you might see familial phenotypes. Whereas when we get up into the space of 1% and above, we go into what we call the common variants. There has been historically a distinction between rare and common, and you will see a number of clinical papers or clinical genetics papers that still try to distinguish them, and it is important. But for this talk and the way we look at it, it's a continuous spectrum. What you're seeing as well, of course, is that when we identify rare variants in this phenotype and the change in menopause age, some of them have very, very high impact. So these are, for example, genes that we recognize clinically, and they can be screened for women with premature ovarian insufficiency or who had mothers, for example, who had an early onset of menopause, that we can go in and we can look at. And we have a list of about 80 genetic variants right now, and they include FSHR, the receptor for SSH, and FOXL2. We also have FMR1. This is a pre-expansion of the FMR1 that we also look at clinically as well, and that most genetic societies and medical reproduction societies suggest that we should screen for. And I will show you later that this is perhaps one of the ones that will be, I think, should be incorporated in greater panels. But then we have at the very kind of bottom right here, all of these common variants that we know a lot of, and that you will see being identified for example, in sequencing studies, but also in genome-wide association studies that I will explain in just a moment. So we're looking at a landscape now of genetic variants and trying to figure out how that, together with environmental factors, shape and determine when we actually go into menopause and we have reproductive senescence. So the human genome contains millions of genetic variants. We have single nucleotide polymorphisms, there are insertion deletions, there are repeat structures as well, as all of you will appreciate. And alleles basically just refers to which version of the genetic variants we talk about. So if it's an A or a T, for example. We have 10 million known single nucleotide polymorphisms in the human genome, and we have at least 10,000 changes to reference genome coding sequence per person, and that's really a minimum estimate. We sometimes see much, much larger numbers than these. So obviously, this is quite a challenge now to figure out how we identify genetic regulation of the phenotype. And are we going to look at many small effects? Are we looking at big effects or combined effects of many different types of impact that genetic variants may have together with environmental factors? So I'm going to focus just on the genome-wide association studies. This is a good place to start. And the reason for that is the UK Biobank is very large, but also because we're looking at a common trait. So that means that it's going to be common genetic variants that's going to shape the distribution on the large massive part of the population distribution. And the way this is done is that here on the left, you see we all, for any given genetic variant, we carry an allele, and we have genotypes. So we might have inherited an A and an A from both of our parents. Sometimes we have a different genotype, a GA, or we have a GG. This is in a case where we have two different alleles at this specific position. We can then go in and we can ask, well, when did menopause age occur in these persons? And then we can then ask, well, okay, how does any given genotype at this specific position, how does that actually contribute in an additive way to the menopause age? And that can both be plus, or it could also be minus, so an extended, or it could also be an earlier onset of reproductive senescence. And we can do this for more than 10 million different genetic variants across the study population that we have. So we've been doing this genome-wide analysis for about 13 million variants with a minor allele frequency that's greater than 0.1%. So we're capturing quite a bit of the genetic variants. And we originally did this in about 200,000 European women or women of European descent. We collected a lot of data from the Reproductive study as well, for 34 studies. We also had interactions with the Breast Cancer Association Consortium for case control studies as well. And then obviously our major source was the UK Biobank. What we found was about 300 or 290 independent genetic signals, and I'm going to get to what that is, associated with age and menopause. So genome-wide association signals, their effects were about 3.5 weeks, so that's quite small impact, but up to about 17 months per allele. So this is quite significant. This is 1.5 years all of a sudden. You have 10 of those, right? And you have 1.5 decades early onset or later onset of menopause. I should also say we then replicated in nearly 300,000 women in 23andMe, and this is really important. And we also did broad replication in about 78,000 East Asian women as well. So this is kind of global regulation we're finding. And there are obviously some minor differences as well, we'll see, because we have different genetic population structures across different ethnic groups. But in general, what we can do is explain about 10 to 12% of variation in age at menopause. And that might sound quite low, but I'm going to get into that. This is actually quite good. So what you're seeing on the graph here is the signals that we see. This is just the volcano plot, and you're seeing the chromosome on the x-axis, 1 through 22, and the X chromosome, and then you're seeing the likelihood or negative likelihood ratio of seeing them. And you can see here that by doing this, of the known ones in red, we knew quite well, and we are replicating them in this much larger study, but we're also seeing a lot of new signals now coming in blue. When we construct polygenic scores from these genetic variants that we have identified from these 100,000 women in UK Biobank with the menopause between 18 and 65, we get genetic risk scores. And I apologize for my voice. I hope it gets better. But when you put the 290 genetic variants together, seen here on the left, you can see here that if we now divide them into the population who had an earlier onset, this is in blue, and in red you see the women with a later onset, more than 45 years. You can see they look quite separate, and that's a good thing. When we then also incorporate a total polygenic score, which we're interested in because we have many more variants and we can impute them, you can see that the polygenic score now, in this case as well, also very nicely is distributed for any individual. And we can look at these effects without and outside the UK Biobank in about 100,000 women that we have done. And on the right-hand side, these polygenic scores, they actually include genetic variants that weren't in the GWAS originally, but we wanted to capture their effects as well. So these might be rare variants as well, for example, from exome sequencing or other sources. So what do we actually see with regards to clinical relevance here? So we have a nice distribution now, but if we now go in and we ask, okay, do these high genetic scores actually correlate, or are they associated with predicting a later onset of menopause in our validation study? And you can see here what we've done is taken the mean genetic score, or rather the standard deviation, and the mean genetic score the mean onset of menopause is around 51, so the genetic variance, the standard deviation there is none. And you can then see that we have a higher genetic score now as women get older in age, and we have a lower genetic score that goes into the negative in the women who are younger, going down to 35. So we're seeing a very, very nice kind of shape that's consistent with the polygenic basis that we've identified in one population is very nice, very good at predicting menopause in an independent population. If there was no prediction, it would just be one flat line around 0. So clinical relevance as well is important because we already now sequence women in fertility clinics who might not be responding and or who have an earlier onset of menopause for the pre-FMR1 mutation, the pre-mutation. And this pre-mutation doesn't make the women ill per se, but it's one of the things that is associated with an earlier menopause age. The odds ratio, if you carry specific pre-mutations as FMR1, is about 5. So you're 5 times more likely than a woman who doesn't carry this to actually go into an early onset menopause at the age of 40. However, only 1 in 250 women actually carry the pre-FMR1 mutation, so it's not a particularly good predictor per se. Whereas obviously for these polygenic scores, all women carry these variants or a combination thereof. And when we go in and look at this, what we find is that if you look at the top 1% of women, so this is 1 in 100 women, or the 99th percentile, the odds ratio of actually experiencing an early onset menopause here now is also in the range of 5, or it's 4.7. This is compared to the 50th percentile, where it's where it's obviously lower. So it suggests to us that with the higher genetic risk, we have more alleles that predispose to earlier menopause, and that's very nice. I should say, though, that one of the things, of course, with signals in genome-wide association studies is they are signals. They're signals that are basically defined by how our DNA is inherited from our parents to child, basically. And that means that there are big chunks called haplotype blocks that get inherited in one go. And then we have to try and see if we can actually tease out what the actual signal or the real true signal might actually be that might be causal for this specific phenotype. And what we do is we go in and we do a consensus analysis, in silico analysis, basically, where we take in a number of different information, for example, about the variants, what kind of variants they are, whether they have an effect on protein structure function, their expression, and also nearest genes as well. When we do this, we found 91 genes that were implicated from variants with functional effects that correlate with these signals. And what we also find is that 20 of the 74 premature ovarian insufficiency genes, they also have a signal nearby as well of the common variants as well. So that's quite reassuring in some ways. And what we identified in the end was about 283 consensus genes. So what we then could do as well was that the exome sequencing from women in the UK Biobank was also becoming available at the same time, and we also incorporated that. And I just want to show you here that by doing this, that we can actually also identify these very rare variants, genetic variants that are important. So if you look here on the x-axis, you can see that we are finding things now that basically are very, very uncommon. That's very towards 0, and then obviously more common ones are towards the right. And the effect now on menopause age as well, we can also see is quite significant. So if you look at these 3 black dots here, one is CHEK2, loss of function variant, which is now basically, if you carry this loss of function variant, you basically have another 3.5 years later onset of menopause. In contrast, if you look at BRCA1 and BRCA2 loss of function alleles, these give you an earlier onset of menopause across genetic background, everything else that one has inherited. I should say as well that this is all for women who are all heterozygous carriers, and these variants are not the ones that are associated with hereditary breast and ovarian cancer predisposition. So how do we try and figure out now as a biological system and how ovarian aging actually works, the establishment as well as the depletion over time? So what we did was we collated and we generated data across the whole lifespan of the cell types, the oocytes and somatic cells before birth, from puberty to menopause, and granulosa cells, to try and look at these signals we have. And in red here, you just see the set score of some genes. You can see they're on very highly, others are lowly expressed in blue. So we see differences at different stages. And what this allows us to do is to go in and start looking at maps of how we can use these genetic signals to actually make pathways. So when we do pathway analysis and we identify the major pathways, we find that DNA repair pathways, so DNA damage sensing and apoptosis pathway, is by far the largest pathway that's coming out. And it's coming out throughout the life course of a female reproductive lifespan. But we also find things that are specific to different stages, like meiotic recombination, which is important, obviously, during meiosis, and is occurring in the fetal oocytes during fetal life. During follicular growth and selection, we've also identified networks that appear to regulate translational regulation, but endocrine metabolic regulation as well. And also one of them is for example, mTOR signaling. We have this, of course, and we can then start making more refined ways of studying and following up on this. And one of the ways we've done this is because the major pathways for DNA repair, DNA damage, sensing, and apoptosis is that we started there. So very briefly, our DNA is constantly insulted with damage to our genome. They include, amongst others, very serious damage such as double-strand breaks, but also replication stress and single-strand breaks that you see on the right here. And we know quite a bit about the different pathways that regulate the detection, the repair, and the subsequent signaling and fate of the cell from the DNA repair field, but also, of course, for cancer, also important in aging. The color code that you're seeing is one that shows that in real or in green are the ones where we detected genetic variants, and you can see that we are detecting quite a few of them. In gray, we didn't detect any genetic variants in this study per se, but we implicated them because of their association in the pathway analysis. So two of the key ones that we were interested in is CHEK2 and CHEK1. These are checkpoint proteins that have previously been implicated in aging phenotypes, particularly for CHEK1. And I will cover these in a second, because what we wanted to do was to go from biological pathway to in vivo models to test whether we could actually extend reproductive longevity. So we have hundreds of mouse models that are infertile. We have 600 of them in total, I think, at the moment. But what we wanted to do was to say, okay, if we go in and start actually perturbing these pathways that we've identified from human, can we now extend reproductive longevity in a mouse model? I know mouse doesn't have menopause per se, but it has a number of features that makes it very good for studying DNA repair, but also ovarian function per se, that I'm happy to cover and take on questions to. So what we did was we went in and we first made the CHEK2 deletion. The CHEK2 deletion, if you remember, this is gene. The actual variant we saw was a loss of function variant that gave women an extra 3.5 years in terms of reproductive longevity. So the CHEK2 deletion we knew is basically fine. They're generally healthy. It's not an essential gene. We knew it's important in oocytes at establishing the ovarian reserve before. To some extent, if you irradiate the fetal ovaries, but on its own, without any other exogenous DNA damage or anything like that, this is just a natural mice in inbred Black 6 background. You can see here in A that the follicle numbers at 1.5 months are very similar between the CHEK2 and litamate controls, whereas when we get to an aged mouse, about 14 months, you can see that we see an increased number of follicles. When we look at AMH concentration, this is very nice as well. This is an aged mouse as well at 16 to 17 months as well. They also have high AMH, circulating anti-Müllerian hormone, which is a hormone secreted by the follicles that are growing. So this is all consistent with that these mice are aging are losing their oocytes at a slower rate than the wild type. We can go in and we can then also ovulate them and treat these mice as if they are actually women, treat them with IVF, just analogous to what we would do with women. So we take a pregnant mare serum gonadotrophin, and we also then trigger with human chorionic gonadotrophin. So this is kind of analogous to what we would do with women in IVF clinics. And this is just the data from the aged mice. You can see here that when we take them, that at 13.5 months, that we get more oocytes out. These are mature oocytes. You can see here that also the proportion of the ones that are fertilized are very similar, and that, again, that the ones that make blastocysts are also similar or not decreased compared to the wild type. So this suggests that the oocytes that are being produced in these aged mice are actually good, they're competent, and there's nothing wrong with them per se. And we can go further down, we can also study that these mice are actually, the offspring are healthy and they're also fertile, which is very, very important for what we do. So we knew for a long time that the other side here, that CHEK1, we didn't identify CHEK1 per se in the actual genome-wide association study, but we were interested in CHEK1 because we found signals that's associated with replication stress and single break repair. And we knew from the literature that an extra copy of CHEK1 can alleviate some features of premature aging in a sickle background. And we were interested in whether this was also the case here, just not in a sickle background, but just on its own. Can an extra copy of CHEK1, can it actually extend reproductive lifespan and longevity in a mouse model? And we basically went in and we first looked at follicles, and you can see here that the CHEK1 mice were born or had a larger number of follicles at 1.5 months, also slightly higher later on. We can go in and we can see that the oocytes that we get out are fine. They're all euploid, which means they don't have chromosome errors. And we can also go in and see that they are actually fertilizable, and that these fertilizable oocytes in an IVF setting also form blastocysts. So this is the kind of thing we would be interested in an IVF setting. So I think just like the CHEK2 deletion mice, loss of function mice, the extra copy of CHEK1, the offspring appear to be healthy and fertile, and we have basically bred them to 4th generation, and everything seems equivalent to those who don't have that extra copy of CHEK1. So what I just want to summarize here is that we've gone from biological pathways to in vivo model of extended reproductive longevity. And what we've done now is use human genetics to inform which mouse models to use, and also to test our human genetics. And just to emphasize here, we have loss of function alleles in CHEK1 that seems to extend reproductive lifespan. In other cases, an extra full copy, functional copy, in this case CHEK1, also seems to do the same in a mouse model. So it seems that different genes and their products and functions are required for different functions as well. So this is quite a refined system. It can also both influence the reserve that's established, but also its depletion over time in the 2 different models. And importantly, when we do this, the offspring are healthy and fertile. So I just want to the last couple of minutes just go in and talk a little bit about the comorbidities, couple of slides that are associated with early or late age and natural menopause. So later menopause is the one that we're particularly interested in, is extending, obviously. And we know that later menopause basically benefits bone health. We also found that it also benefits type 2 diabetes risk as well. It's below 1, as you can see here. But having a later menopause also increases the risk to hormone-sensitive cancer, estrogen-sensitive cancers as well. And you can see here, these include endometrial cancers, breast cancers, and ovarian cancers. Although you can see that the odds ratios to some extent are quite modest. So I just want to summarize here and say that what we've done is gone from life course to molecular mechanisms. We obviously just shown the proof of concept, and there's just so much to unravel to try and understand what determines this really elegant but important biological system. And then the final perspective here I have is simply that we have not been able to study a lot of the genetic variants or gene functions even in the system because it's required in vivo model. We haven't had any cell lines. But a breakthrough from 2 Japanese groups has really now allowed us to do this and generate and look at all of the entire life cycle of oocytes and granulosa cells in vitro in these organoid systems. So with that, I obviously hope that we can go and we can basically start doing a lot more studies to try and understand this beautiful system. Thank you very much.
Thank you so much. What a great consortium you have with so much power, I would say, and so many data. I was quite surprised, I must say, that 10% of the babies born in Copenhagen, in Denmark, are actually the birth is based on medical interventions. So yeah. Is that number rising? So can we call infertility a modern disease?
I think so. And I think WHO has also called it that. I think the proportion really depends on the availability. And in Denmark, it's free at the point of care for women up to 42. for 3 cycles, for example, right? But that's only for primary, but they're talking about increasing that, and also both the number of cycles and also for secondary infertility as well. There are other countries that have similar features with regards to medical interventions, Israel, for example, and there's no restriction on the number of cycles that you can receive treatment for. Other countries like other countries where it really depends, the UK, for example, has a little bit of a lottery code where you live, whether the local and trust will pay for fertility treatment, particularly if you're a bit older, advanced maternal age.
Yeah. So, and if I understood correctly, then the fertility period is not really following the longevity trait we actually see. So, and that brings me to the next question. How much of the aging process we know of, how much is related or is a little bit transferable to ovarian aging?
Yeah, that's really a great question. We don't really know right now whether some of the signals will be shared. We're looking at that. We don't have a lot of information on fertility. And the reason for that, the fertility curve or the aneuploidy curve, and the reason for that is that measuring aneuploidy in human eggs is very tricky. But there are ways of doing it now that I'm hoping that with the data availability, we'll be able to run genome-wide association studies and also understand that more and see if there's overlap between the two.
So, and as you know, as you're working with the UK Biobank, we just were working on the UK Biobank, looking at the rate of aging of different organ systems, but excluding ovarian. So, yeah. Do you have any idea or a sense of the rate of aging of different organ system in these women is actually correlated or associated with the ovarian aging? So, is there a trait which is not ovarian specific?
Yeah, so John and Anna, they are looking at that at the moment with their colleagues to try and figure out are there any specific features that co-vary with the ovarian aging. I think it's a very, very important question, interesting question as well. I mean, we know features such as, for example, clinical diagnosis of osteoporosis, but fractures as well obviously also goes down if you have a later onset of menopause. So there's certain, so I guess that certain systems should be coming up if you did the analyses.
Following up on that, you have all these very nice GWAS studies in the SNPs. Have you actually looked in the SNPs who are contributing to 10 to 15% of the menopause or the age of menopause, if these are ovarian specific or also associated with other, maybe an absence of disease or aging phenotypes?
Oh, this is really great. Yeah, we're doing that at the moment to try and figure that out. The one of the big features, obviously, is that we're looking at DNA repair and DNA damage response, and they are important for many processes, right? So I think that it's going to be very, very interesting what we see. One thing I can say from Stasia Stankovic's work that we have now deposited on medRxiv is that cardiovascular disease doesn't seem to come up very strongly at all as a comorbidity with ovarian aging, whereas, there's been tremendous focus on that women go into menopause. It's all hormonal with cardiovascular phenotypes later in life. And we don't really see that signal in there.
Yeah.
Or the association between the two.
Okay. Okay. So, and you already argued, look, it's a lot, 10 to 15% of variation contributing to the age at menopause. And I think you are right, because if we are looking at the literature, the height, I think is the best one was 5% or something like that. So well done. What are the other contributing factors? And maybe then also closing the loop a little bit, like infertility a modern disease? So how much of the variation can you actually then contribute to other factors like a higher BMI, etc.?
This is great. I think that smoking is the one we know the most about because it's been studied quite extensively. What we find is that when you look at the area under the curve of the polygenic risk score that we have, we find that you can explain that we explain about 66%, so 0.66, which is modest. It's not great. Clinically, you need to get up to 0.8 for the polygenic risk score we have. Smoking itself, it's better than smoking though, because that's only at 58%. if you put our polygenic risk score together with smoking, there's no added value to it, suggesting that smoking might be acting through the variants that we have identified, which kind of makes sense because DNA repair and DNA damage response is so prevalent. I think that the closing with fertility, importantly, smoking is not important for infertility per se, certainly not with regards to the fertility curve and aneuploidy, the incidence of chromosome errors in human X that will lead to, for example, Down syndrome. That's been studied quite extensively in the US National Down syndrome cohort, that smoking per se is not a major factor in the risk, for example, of conceiving a trisomic 21 as women age. So I think we are looking at potentially two different systems with genetic regulations or regulators in there as well. And I think that I would like to say that we can do more, but I mean, for example, BMI is obviously a very important factor with regards to also response to stimulation in IVF clinics. That's why across the world, there's generally speaking a limitation to when, with the BMI and then treatment, for example. And weight loss is an important aspect, not just for fertility, but also subsequent pregnancy.
So also here for ovarian aging, lifestyle is of utmost importance, and do not smoke.
Yes.
Yes.
Yeah.
Okay. Let's go to the questions of the audience. I would like to introduce Dong Lu, who's a PhD student, and I think the topic is dear to her heart. So Dong, the floor is also yours. What does the audience have to question and to ask?
Thank you, Andrea, and thanks very much, Prof Hoffmann, for the illuminating talk. We've got a lot of questions from the audience. I myself worked on ovarian aging and do find some of these questions interesting and important. Now I'm going to start with some general questions from the audience. It seems like many of our audience are interested in factors driving ovarian aging, such as smoking, as you mentioned. So we have a question from Mohan. He was asking, what are the dietary factors affecting the aging of oocytes, if there's any?
Yeah, diets is an interesting one. BMI is definitely a factor, but diet itself is very unclear. Should we put it that way? We don't really see a huge association, for example, with lipids, circulating serum lipids. So that's perhaps in the UK Biobank in our subsequent study. I don't really want to start guessing on nutritional value. What I can say is the following. There has been a number of studies done on oocytes aging with regards to their genetic quality. So this is aneuploidy, so chromosome errors that come in and that usually result in infertility, i.e. that the pregnancy goes to a pregnancy loss. So 25% of pregnancies are lost. So when we look at aneuploidy in human eggs, we know that, for example, if you now do in vitro maturation of the egg, so that's when the chromosomes come apart, if you do that in vitro, you can use coenzyme Q10 and you see a difference. You see about a significant improvement in reduction in chromosome segregation errors and subsequent aneuploidy. Now, when this was trialed in a prospective RCT, they didn't see an effect. It was slightly underpowered, and I think another issue, of course, was that the coenzyme Q10 might not actually have been bioavailable either. That could have certainly have explained it. But certainly, I think that there are ways to start doing interventions with nutrition, certainly for the specific part of fertility, whether there is for menopause as well over a short period. I don't think I'm comfortable commenting on. I think a healthy lifestyle and a health span, generally speaking, is a good thing, right?
Yeah, it seems like whatever you do for the general aging is good for the ovarian aging process.
Yeah.
We have another question from David. It seems like he's a bit worried because he asked, does the age at which the first pregnancy occurs impact the subsequent rate of ovarian aging?
We don't have data that suggests that's the case, actually. I don't think that we have now data that suggests that the age at puberty in some cases is linked to when you go into menopause, right? But again, in some women you see it, in others there's no coupling. But we've now started seeing coupling in the signals in the UK Biobank between the start of the reproductive phase and when it ends.
I see. So
I should say there's the only risk factor we know of the first early pregnancy is the age of early pregnancy. We know that if you have it as a young teenager, for example, then you have some kind of protection against breast cancer later in life. And we don't understand how that works. But yeah, that's quite well documented epidemiologically.
I do came across a paper that shows a correlation between late pregnancy and ovarian longevity. And I think there's something behind it that may need more investigation. And now I'm kind of going to the next topic. So Andrea and you were talking about specific features of ovarian aging that are different from the general aging process. It seems that our audience are curious about reasons behind this uniquely early and fast aging rate of ovaries. So we have a question asking, why does the female reproductive system age faster than most of the other organ systems within the female body? I think it's a trick questions to answer.
Well, we don't have stem cells to regenerate. That's really important, right? So unlike the testes, you have spermatogonial stem cells. There's a seeds that will regenerate over time. Whereas in the ovarian system, you are born with a ovarian reserve. The vast majority of mammals are, and they're then depleted over time. So there's no regeneration per se in the ovary. So that's certainly one explanation why we have a defined endpoint or the acceleration in terms of ovarian aging.
May I just ask you about, there are other systems in the human body where we don't have the stem cells. Think about the neural stem cells. So what is so different with the ovarian?
So the ovarian, I think, to some extent has been selected with regards to the distribution that we see. When you have your children, it's part of our evolutionary life history. Specifically, if I now focus on the fertility curve, but it's the same with also with the menopause curve. It's been suggested from anthropologists that this is a selection for the lifestyle and the sociological factors that are driving that have been driving and putting selective pressure on the biological system to some extent. So, for example, when you look at the age of onset of menopause, it's generally when the females, your daughters, are about to have their children. And we have a very strong hypothesis that suggests that with evidence, that if you have a grandparent, a grandmother specifically, that you're then more likely to have grandchildren. So the idea has been that females, they stop having their own children and invest in their children's children instead as they grow older.
The grandmother hypothesis.
Yes.
Yeah, this is called the grandmother hypothesis. That's the actual name for it. Yeah, and there's a lot of both extant data and also historical data that's the case.
That's very interesting. Thank you, Prof. Hoffmann. And we have a question from Zhong Wei. He's actually the Assistant Director of NUS VIA Co-Asian Center for Reproductive Longevity and Equality. He raised an important point here. He mentioned that ethnic differences do exist for ovarian aging, especially among Asian women, which the data is quite lacking currently, versus a woman of other ethnicities. And this really needs to be examined separately. And this really calls for collaborative efforts to understand more generic biological mechanisms. He raised a question about marker of ovarian reserve, which other audience seem to be interested as well. Yeah. He mentioned that AMH, the anti-Mullerian hormone, seems to be the only marker for ovarian reserve used clinically. Will there be a time when utilizing a typical genetic expression profile based on, for example, GWAS and mechanistic studies, which is ethnic specific, individualized and precise prediction of ovarian lifespan, and hence this can be used to determine women's healthy longevity in post-reproductive years?
Yeah, so there are two parts to this. There's the actual measurement or clinical assessment of ovarian reserve, and then obviously whether that predicts post-reproductive longevity and health subsequently. So starting with AMH is currently used, that's correct, clinically. Obviously, we would like different markers. Usually the different markers we incorporate is family history. So did your mother have an early onset menopause? And also the women that you might see in clinics very early on being actually looked at, particularly those at very early onset menopause, before 40 and go into the premature ovarian insufficiency clinical group. So I think, yes, we want to incorporate polygenic risk scores as we know them, but also obviously as we expand that into exome and whole genome sequencing, any rare variants as well that we can actually detect and put it in together with lifestyle factors, his family history, etc., to get a refined way of looking at it. I mean, AMH as well, as I'm sure the professor also is aware of, and I think most of us are aware of, is highly variable between cycles as well, between, in the same woman. And it's produced in the small growing antral follicles, right? So it's an average what you're seeing in a serum level, but if you also have differences in the follicles, which we know we do, it's not that accurate.
Based on that, if we can have really individualized prediction for the reproductive life, could some of the early menopause phenotypes due to these genetic variations be beneficially impacted by specific or individualized pharmacological, nutritional, or lifestyle interventions?
I'm not sure about lifestyle interventions. I want to be very, very careful with that because I think we need evidence to look at that. It's also obviously, I think we're dealing with the reproductive system, so we would need to figure out as well with regards to what comes later, and that's obviously the fetal health and children's health.
Okay, last burning question, very short answer. Do we have one?
There's actually a question that is specific to what you have presented. So he was asking, since the deletion of CHEK2 and extra copies of CHEK1 are both linked to decreased ovarian aging, this audience was wondering if intervention on both would lead to synergistic effects.
Yeah, we're looking at that at the moment. Yeah, it's taking away its aging, right? So yeah, we're looking at that at the moment.
Look forward to that piece of work in the future.
Thank you.
That was a very short answer. Thank you so much. I think we learned so much about genetics contributing to menopause, etc. Just 10 to 15%, and I think that we shouldn't underestimate our lifestyle, which is, I think, very important also for other aging processes in our organs. So thank you so much, Eva, for this great presentation. Thank you.
Thank you for having me.
I think this presentation triggered lots of questions and maybe also comments. So please, as always, use the chat function using the panelists and attendees options to leave your feedback. Also, if you have other burning questions, please leave them there and we will try to answer them. Once again, I would like to remind you, please do not miss the opening of the Centre for Healthy Longevity at the 7th and the 8th of September. And there is the link in the chat box, how you can register. Once again, at the 8th, we will not have a webinar, but we will have a webinar, which I will host at the 11th of August. And there we will talk about aging, Alzheimer's disease, and artificial intelligence. And the speaker will be Associate Professor Evandro Feng, and I'm very much looking forward to that. I will close this session with a video, which is much related to this evening and to this great presentation, showing and highlighting a woman being a mom at old age. Take care.
There's so many benefits for waiting. Being older, I am more confident in who I am as a person, as a woman. I just have so much more life learnings that I can bring to the table. I just feel way more prepared. I think us women are way more concerned about education and setting a solid career path. It's really unfortunate, I feel, that we have to almost decide, well, I'm gonna forgo furthering my career because I wanna have children, or I don't have this career because I wanted children. There shouldn't be a stigma for older women. I just think that anyone could have an issue, not just based on age. When I look at Maxwell, I kind of look at her in disbelief. I cannot believe that we created her and created somebody so strong. I also think about all the trials and tribulations and all the hardships that I've went through, realizing that like all these things had to happen for her to be here. And I feel like things happened exactly the way they were supposed to happen.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.